Welcome everyone. I'm excited to kick off our presentation today. We'll be exploring the fascinating world of energy-rich compounds. The title of our talk is Energy-Rich Compounds, and we'll also look into some key concepts and examples. Over the next few slides, we'll cover what these compounds are, why they're important, and how they function. Let's get started. Organisms require energy to carry out a variety of activities, such as muscle contraction and other cellular movements. This includes processes like active transport and the synthesis of macromolecules. All these activities are quite demanding in terms of energy, and they rely on chemical energy to power them. Chemical compounds store this energy, and they release it through a process called hydrolysis, where high-energy bonds within these compounds are broken. When these bonds are broken, energy is liberated and becomes available for the cell to use. This is the fundamental theory behind how energy is transferred and utilized within living organisms. Understanding this process is essential because it underpins how cells perform their vital functions efficiently and sustainably. When hydrolyzed products go energetically low, meaning their Gibbs free energy change is negative, they release energy. This is a key concept because it explains how energy is made available in biological systems. Now, let's talk about high-energy phosphate compounds. These are molecules that contain phosphate groups and are considered high energy if their standard free energy change for hydrolysis is more negative than about 20 to 25 kilojoules per mole. This negative value indicates that breaking these bonds releases a significant amount of energy that can be harnessed by the cell. It's important to note, though, that these high-energy phosphate compounds are not meant for long-term energy storage. Instead, they serve as temporary energy carriers, kind of like a rechargeable battery, that transfer energy quickly from one reaction to another. This rapid transfer is essential for maintaining efficient cellular functions, especially during processes that require immediate energy input. So, in the broader context of our presentation, understanding these compounds helps us appreciate how cells manage their energy needs dynamically, using these high-energy molecules as quick, short-term energy reservoirs to keep everything running smoothly. Let's talk about endergonic reactions. These are reactions that require energy input to proceed. Examples include active transport, which helps move substances across cell membranes, muscle contraction, essential for movement, and the transmission of nerve impulses, which allows signals to travel through the nervous system. Aside from ATP, which you're all familiar with, GTP or guanine triphosphate also plays a crucial role as an energy source. It's particularly important in processes like photosynthesis, where new proteins are made, and gluconeogenesis, the creation of glucose from non-carbohydrate sources. Additionally, UTP or uridine triphosphate and CTP or cytidine triphosphate are used as energy sources in the metabolism of carbohydrates and lipids, respectively. These molecules help drive various metabolic reactions forward. Now, regarding free energy, ATP serves as the principal immediate donor of energy within cells. It's like the cell's primary currency for energy transfer. There are 5 main types of high-energy bonds in molecules like ATP. The most important ones are phosphoanhydride bonds, which are formed between 2 molecules of phosphoric acid. For example, in ATP, these are the 2 high-energy diphosphate bonds. It's worth noting that the bond between the phosphate and ribose in ATP isn't as energy-rich as the phosphoanhydride bonds. That's because it's an ester bond, which doesn't store as much energy. Understanding these high-energy bonds helps us see how energy is stored and released in biological systems, which is fundamental to cellular function and overall metabolism. Let's take a closer look at some key energy-rich bonds in biological molecules. First, we have the enolphosphatic bond. This bond is actually quite high in energy. Its hydrolysis releases about 61 kilojoules per mole. You'll find this bond in phosphoenolpyruvate, which plays a crucial role in glycolysis, specifically during the breakdown of glucose. Next, there's the acyl phosphatic bond. While not as energetic as the enolphosphatic bond, it still releases a significant amount of energy, around 49 kilojoules per mole upon hydrolysis. This type of bond is present in 1,3-bisphosphoglycerate, another key intermediate in glycolysis. Moving on, we have guanidine phosphate. This bond forms when phosphate attaches to guanidine, and it releases about 43 kilojoules per mole when hydrolyzed. A good example of this is in phosphocreatine, or PC, which is found in muscle cells. It acts as a quick energy reserve in tissues, especially during intense activity. Finally, there's the thyristor bond. Unlike the others, this bond isn't as high in energy because it doesn't contain a rich phosphate group. Nonetheless, it's important. It's present in molecules like acetyl-CoA, which plays a central role in metabolism, especially in energy transfer and synthesis processes. Connecting all these, it's clear that different bonds release varying amounts of energy, and their presence in molecules like glycolytic intermediates or energy reserves highlights their importance in our overall energy management. When we look at various high-energy bonds, we're essentially exploring the different types of bonds that store a lot of energy and can release it rapidly. These bonds are crucial because they act as the energy currency in many biological and chemical processes. You might be familiar with some common examples like ATP, which is often called the energy currency of the cell. But there are also other forms, such as certain chemical bonds found in fuels or explosives, that have high energy content. Understanding these bonds helps us grasp how energy is stored, transferred, and utilized in various systems. Throughout this presentation, we'll see how these high-energy bonds play a vital role in everything from cellular function to energy production in industry. Recognizing the characteristics of these bonds also allows us to compare their stability and energy release, which is key for both scientific understanding and practical applications. Let's take a closer look at how ATP works. ATP, or adenosine triphosphate, is often called the energy currency of the cell. Essentially, it stores energy in its high-energy phosphate bonds. When the cell needs energy, for example, to contract a muscle or to power a biochemical reaction, ATP is broken down into ADP, or adenosine diphosphate, and an inorganic phosphate. This breakdown releases energy that the cell can then use for various functions. Think of ATP as a rechargeable battery. When the cell has excess energy, say, from food intake, it recharges this battery by adding a phosphate group back to ADP, turning it into ATP again. This cycle of breaking down and rebuilding ATP is at the core of how cells manage energy efficiently. Understanding this process helps connect to the bigger picture of metabolism and how our bodies sustain life through continuous energy transfer. It's a simple yet incredibly elegant system that keeps everything running smoothly. The phosphate groups in ATP are connected by very high-energy chemical bonds, which means these bonds store a significant amount of energy. When the cell needs energy to perform work, these bonds can be broken under certain conditions. Specifically, the end phosphate group can break away from the ATP molecule, releasing that stored energy. This released energy is then used to power various energy-hungry reactions within the cell, helping to keep the cell alive and functioning properly. Understanding these high-energy bonds is crucial because they are at the core of how cells store and transfer energy efficiently. When the end phosphate is released, what's left is ADP, or adenosine diphosphate. This change from ATP, which has 3 phosphates, to ADP with 2 phosphates, is happening constantly as ATP circulates through our cells. It's a really dynamic process, and it's essential for providing the energy our body needs for various functions. Recharging ADP back into ATP requires a significant energy investment, and that energy comes from the food we eat. So, in a way, our food fuels this cycle, allowing our cells to keep working efficiently. This continuous cycle of breaking down and recharging ATP is at the core of how energy flows within our bodies. Now, let's look at adenosine diphosphate, or ADP, and compare it to adenosine triphosphate, ATP. As you can see, ATP has 3 phosphate groups, indicated here by the Ps. These high-energy phosphate bonds are crucial for storing and transferring energy in cells. When ATP undergoes hydrolysis, which simply means adding water, it breaks down into ATP and an inorganic phosphate. This reaction releases energy, and we call it exergonic, meaning it releases energy that the cell can then use for various functions. So, in essence, breaking down ATP into ATP and phosphate is a key step in providing the energy necessary for many cellular processes. This process ties directly into the overall theme of how our cells manage and utilize energy efficiently. Now let's take a closer look at how ATP is formed from ATP. As you can see here, ATP, which stands for adenosine diphosphate, combines with an extra phosphate group, represented by the P. When this happens, we get adenosine triphosphate, or ATP, along with a water molecule. This process is known as dehydration synthesis because it involves removing a water molecule to join the molecules together. It's important to note that this reaction is endergonic, meaning it requires energy input to occur. Think of it as storing energy in the bonds of ATP, which is essential for powering various cellular activities. So, when ATP gains that phosphate, thanks to dehydration synthesis, it transforms into ATP, the cell's primary energy currency. This process is fundamental to how cells store and utilize energy efficiently. Now, let's talk about high-energy bonds, which are represented by this tilde symbol. These bonds are found in molecules like ATP, and they play a crucial role in energy transfer within cells. Specifically, the tilde P here stands for a phosphate group that's attached via these high-energy bonds. When we hydrolyze these bonds, it releases a significant amount of energy, indicated by a large negative delta value. This large negative delta means that the process is highly favorable and releases energy that can be used for various cellular activities. So, in essence, ATP acts as the cell's energy currency, thanks to these high-energy phosphate bonds that readily break to fuel biological processes. Understanding this helps us see how energy flows through living systems, powering everything from muscle contractions to nerve impulses. All right, let's break down what's happening with ATP and its related molecules here. First, we see that ATP can be broken down into ATP and inorganic phosphate, pi. This is a key step in providing energy for cellular processes. Then, ATP itself can be further broken down into AMP and pi, which essentially repeats the cycle of energy release. Now, there's an alternative pathway where ATP directly converts to AMP and pyrophosphate, PPI. This shows how versatile these molecules are in different biochemical contexts. When PPI is produced, it can then be split into two pi molecules, completing that part of the process. Moving on, AMP can also be converted into a diphosphate form, AMP P tilde P, which can then be broken down into AMP with PP, or pyrophosphate, and finally into AMP plus pi. The important thing to remember is that these steps are reversible, and they're crucial for maintaining the energy balance in the cell. And finally, when we see P tilde P, or pyrophosphate, it can split into two pi molecules. This dissociation process is vital because it drives these reactions forward and helps regulate the overall energy flow within the cell. So overall, these reactions highlight the dynamic nature of nucleotide metabolism and how energy is stored and released through these interconnected steps. ATP is often called the energy currency of the cell, and for good reason. It's the molecule that powers almost all cellular activities. When we talk about ATP, we're really talking about how cells store and transfer energy efficiently. Every time your muscles move, your brain processes thoughts, or your cells build new molecules, ATP is involved. It's like the battery that keeps everything running smoothly. Without ATP, our cells couldn't perform their essential functions, and life as we know it would grind to a halt. So, understanding ATP helps us grasp how energy flows through living systems and why maintaining this energy balance is so vital for health and survival. Now, let's take a look at phosphorylated compounds, which play a crucial role in energy transfer within the cell. One key example is thioesters, such as acetyl-CoA. these compounds are particularly important because they have a large free energy change associated with their formation and breakdown. This means they can store and release energy efficiently, making them vital players in metabolic reactions. Understanding these compounds helps us see how energy flows through different biochemical pathways, tying into the broader theme of how cells manage and utilize energy effectively. Let's take a closer look at some key phosphorylated compounds involved in energy transfer and metabolism. First, phosphoenolpyruvate, or PEP, is a high-energy compound that plays a crucial role in glycolysis, acting as an energy donor for ATP synthesis. Moving along, 1,3-bisphosphoglycerate is another high-energy intermediate, important in glycolytic pathways. Then we have phosphocreatine, which serves as a rapid reserve of high-energy phosphate groups in muscle cells, helping us regenerate ATP quickly during intense activity. Speaking of ATP, it's the primary energy currency of the cell, fueling countless biological processes. ADP and AMP are its lower-energy forms, and their ratios help regulate energy status. Pyrophosphate, or PP, is a byproduct of biosynthetic reactions and can influence the direction of certain metabolic pathways. On the carbohydrate side, glucose 1-phosphate, fructose 6-phosphate, and glucose 6-phosphate are all key intermediates in carbohydrate metabolism, especially in glycolysis and glycogen synthesis. Lastly, when we refer to phosphorylated compounds overall, we're talking about molecules that contain phosphate groups, which are essential for energy transfer, signal transduction, and metabolic regulation. Connecting these points back to our overall theme, understanding these compounds helps us appreciate how energy flows within cells and how metabolic pathways are tightly regulated. Let's take a closer look at phosphoenolpyruvate. It contains a phosphate ester bond, which is key to its reactivity. When this bond breaks, it transforms into the enol form of pyruvate. Now, the interesting part is that the enol form isn't very stable on its own. However, it can rapidly tautomerize into the keto form of pyruvate, which is much more stable. Because phosphoenolpyruvate exists primarily in this enol form, but pyruvate can adopt two different forms, the enol and the keto. The overall process favors the formation of the more stable keto form. This stabilization of the product relative to the reactant significantly contributes to the high negative free energy change observed during hydrolysis, which is about -61.9 kilojoules per mole. This large free energy release is a fundamental aspect of why phosphoenolpyruvate is such a high-energy compound. It's this drive toward the more stable keto form of pyruvate that makes its hydrolysis so energetically favorable, and it's a key step in metabolic pathways like glycolysis. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's take a closer look at 1,3-bisphosphoglycerate. This molecule contains an interesting feature, an anhydride bond between the carboxyl group at carbon 1 and a phosphoric acid group. This bond is quite significant because it's a high-energy linkage. Now, when we consider the free energy change, delta-0, for reactions involving this molecule, we see it's very large and negative. This large, negative delta-0 essentially means the reaction is strongly favorable and can proceed spontaneously. The key to understanding this lies in the structure of the reactants and products. The high-energy bond in 1,3-bisphosphoglycerate is a major contributor to this energy release. Connecting this to the overall theme of energy transfer in metabolism, molecules like 1,3-bisphosphoglycerate act as energy donors, helping drive essential biochemical reactions forward. So, recognizing the structural basis of these energy changes helps us understand not just this specific molecule, but the fundamental principles of energy flow in biological systems. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's take a closer look at phosphocreatine. In this molecule, the key point is the PN bond, which can be hydrolyzed, that is, broken down by adding water, to produce free creatine and inorganic phosphate, or PI. Now, this hydrolysis is important because the release of pi actually makes the reaction more favorable. Plus, creatine benefits from resonance stabilization, which further pushes the reaction forward. This process is a crucial part of how our muscles rapidly regenerate ATP during short bursts of intense activity. So, understanding how phosphocreatine works helps us see how energy is stored and quickly mobilized in our bodies. This slide doesn't contain enough information. That's why we're moving to the next slide. Thywesters have a large, negative standard free energy change of hydrolysis. This means they are quite reactive and tend to break down readily, releasing energy in the process. Acetyl coenzyme A is one of the most important thywesters in metabolism. It plays a central role because the acyl group in these compounds is activated, making it ready for various reactions like transacylation, condensation, or oxidation reduction. Essentially, these thywesters act as energy-rich intermediates, facilitating many vital biochemical transformations. Their high reactivity is key to enabling the efficient transfer of acyl groups, which is fundamental to metabolic pathways. Recognizing their importance helps us understand how cells harness energy and build complex molecules efficiently. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about NAD+, which stands for nicotinamide adenine dinucleotide in its oxidized form. This molecule plays a crucial role as the major electron acceptor in catabolic reactions. It's quite powerful. It can actually oxidize alcohol groups to form carbonyl groups, which is a key step in many metabolic pathways. NAD+ is essential in processes like beta-oxidation, glycolysis, and the TCA cycle. Without NAD+, these pathways simply couldn't proceed, which highlights just how vital this molecule is for energy production and overall metabolism. Now, when NAD+ accepts electrons, it becomes NADH, its reduced form. Think of NADH as carrying a load of electrons, specifically in the form of hydride ions. This molecule then donates those electrons to the electron transport chain, or ETC, helping to drive oxidative phosphorylation, our main way of producing ATP. NADH also plays a role during fermentation, donating electrons to convert pyruvate into other products, depending on the type of fermentation happening. And there are other related compounds involved in these processes, but the key takeaway is that NAD+ and NADH are central players in energy metabolism, acting as electron carriers. They link the breakdown of nutrients to the production of usable energy, keeping our cells functioning smoothly.